Hi, my friend. Nice to see you again. Do you remember who I am? I am a neuron. Last time I talked about my basic structure and components. Today I will talk about the way I find and communicate with my peers. Let me introduce my partner, Neurorange and Musmint. Neurorange is a neuron 2 and Musmint is a muscle cell. To communicate with them, the first step is to find my way. It is achieved through axon guidance. In general, in the development stage, my growth cone receives some attractive or repulsive extracellular cues. When the signal is transduced into my body, the cytoskeleton dynamics changes, thus results in growth cone turning via filopodial withdrawal or dilation. There are two types of signal, short range, which are surface protein or signals that are secreted but not diffuse, and long range, which are secreted and diffuse. Here is an example. Imagine that I am an olfactory receptor neuron, short for OR. Our cell body locates in the olfactory epithelium, while our axons project to the olfactory glomerulus in the olfactory bulb. Look at the graph on the right. It shows the map of OR neuronal projections. You may notice that the axons are arranged in a linear manner. Why? You may remember that we've talked about cell signaling in the last video. Here it is again. Olfactory receptors are kind of G protein coupled receptors. Ligands, in this example, the chemicals received by the olfactory receptor, binds to the GPCRs, thus activates G proteins. G proteins activate AC3, turning ATP into cyclic AMP. Each OR generates a unique level of CAMP signaling. CAMT activates pKa. And pKa catalytical subunit phosphorylate CAMP response element binding protein or CREB. Phosphorylated CREB bind to CRE on the gene sequence and regulate gene expression. Since cyclic AMP level is unique of each kind of receptors, gene expression is different for each of them too. High cyclic AMP level results in high neuropilin-1 level and low semaphorin 3A level, and vice versa. Semaphorin 3A is a repulsive cue to the receptor neuropilin-1. As you can see in this graph, semaphorin 3A and neuropilin-1 both locate on OR axons. Different neurons have different gradients of SEMA3A and neuropilin-1. When projecting to olfactory glomerulus, neurons that have higher SEMA3A level goes to the anterior part, while those have higher neuropilin-1 level goes to the posterior part. Since axons repel each other, the map is discrete. As you can see, through axon guidance, I found my friend, high Neurorange. My growth cone attached to its dendritic spine. The next step is synaptogenesis, to generate a synapse. There are 4 stages. First, recognition stage, which involves cell-to-cell recognition and adhesion. Adhesion factors like cadherin homodimer are required in this stage. Second, formation of tight junctions between presynaptic and postsynaptic cells. Third, the maturation of extracellular matrix. And last, vesicles cluster in the presynaptic membrane. It's my membrane, by the way, forming the active zone, while neurotransmitter receptors cluster at the postsynaptic membrane. In stage 3 and 4, inducive factors, ephrin B, FB, neuraxin, and neurogelin, scaffolding proteins, are involved. Since the synapse between Neurorange and I is a neuron-neuron synapse, glutamate is used as a neurotransmitter. If the synapse is between me and Musmint, a neuromuscular junction, things would be a little different. Acetylcholine is used as neurotransmitter, while agarine and musk are essential inducing factors. In inhibitory synapse or the GABAergic synapse, GABA is used as a neurotransmitter, while gafferin, a scaffolding protein, cluster GABA and glycine receptors. After the synapse is formed, you may be interested in the way we talk to each other is through synaptic transmission. First, neurotransmitters are synthesized and stored in the vesicles. Next, AP invades at the presynaptic terminal. Next, there is the opening of the voltage-gated calcium ion channels, and then there is a calcium ion influx. The calcium binds to the synaptotagmin, which catalyzes fusion between the vesicular membrane and the plasma membrane. Neurotransmitter release into the synaptic cleft via exocytosis. Then there is neurotransmitter bind to the receptors on the postsynaptic membrane, which results in the opening or closing of the postsynaptic channels. Also, the postsynaptic current results in the excitatory or inhibitory postsynaptic potential that changes the excitability of the postsynaptic cell. Then there is the removal of neurotransmitters by reuptake or enzymatic degradation. At last, there is the retrieval of vesicular membrane from plasma membrane. At first, the clustering triskidia coated and then the dynamic pitch of the vesicles from the membrane. What about the potential on the postsynaptic membrane? Let's use neuromuscular junction as an example. One vesicle release will result in one miniature end plate potential on the postsynaptic membrane. When there is a stimulus on the presynaptic membrane, calcium ion flow into my body via voltage-gated calcium channel, which results in the release of a number of vesicles. As a result, a number of MEPPs accumulate at the postsynaptic membrane, which leads to a bigger end plate potential. How information is encoded through neurons. Let's talk about the synaptic plasticity. First, the short-term synaptic plasticity. The first stage is called facilitation, when there is a series of high-frequency stimulation, presynaptic calcium ion buildup bind to synaptotagmin and leads to more neurotransmitter release to the synaptic cleft. Then it comes depression. Since the number of vesicles in the readily releasable pole is limited, there is vesicle depletion. Then there is augmentation. The prolonged increase in the presynaptic calcium enhances the activity of the MUNC13, thus enhances the ability for calcium to trigger synaptic vesicle fusion. It causes the rise and fall of neurotransmitter release in a few seconds, making response bigger in scale. At last, in a time scale of 10 seconds to minutes, there is potentiation. The prolonged increase in presynaptic calcium ion makes synapsin dissociate from the vesicles, thus release additional vesicles from the reserved pool. Then let's talk about the long-term synaptic plasticity. As you can see in the green graphs, if you keep touch the siphon of an aplasia, the magnitude of gill contraction will keep decreasing. It is called habituation, whose mechanism is also the reduction of synaptic vesicles available to release, but in a longer timescale. Also, in the purple graph, if you provide a shock to the tail and then touch the siphon, there will be a more significant response of gill contraction. This is called sensitization, which involves the recruitment of additional neurons. As you can see in this graph, in normal withdrawal reflex, only the blue sensory neuron on the top, the interneuron and the motor neuron are involved in the circuit. However, in a sensitized withdrawal reflex circuit, the sensory neuron innervating the tail and the modulatory interneuron are also involved. How does the modulatory interneuron, also called the facilitatory interneuron, work? The mechanism for short-term sensitization and long-lasting sensitization is slightly different. For short-term sensitization, as you can see on the top, it depends on the pKa-dependent enhancement of glutamate release from the presynaptic membrane of the sensory neurons. For long-lasting sensitization, persist pKa activates CREB, which results in changes in gene expression via transcriptional factors. This kind of sensitization is especially important in human hippocampus tree synaptic circuit, which might be essential for learning and memorization. During development, the physiology of neuron circuits should be modified depending on the individual's activity. The process is called synapse refinement or pruning. More specifically, pruning is defined as selective remove experienced neuronal branches and connections in the immature system to ensure the proper formation of functional circuit. There are three kinds of synapse refinement: refinement of topography, refinement of convergence, and refinement of postsynaptic component. Synaptic refinement is necessary for the proper function and structure of neuronal systems. Let's take mammalian olfactory system as an example again. In the first part, I talk about the pre-sorting of olfactory neurons dependent on the stochastic CMP level. On the contrary, pruning is dependent on the local and competitive odor-invoked neuronal activity. As you can see, the left diagram shows the normal pattern of the olfactory glomerulus. Olfactory neuron axons of the same time form homogeneous synapses. However, the right diagram reveals heterogeneous synapses. This indicates that there is something responsible for the generation of gathered axons. It is the random inactivation of CNGA2 channel. CNGA2 plus X chromosome results in chiral-2 expression and high afferent A level, while CNGA2 minus X chromosome results in chiral-3 expression and high Fa level. Chiral homophilic adhesion and Fa repulsion finally leads to the unique distribution of the axons. Okay, this is the way to build a neural circuit. Thanks for watching. May the healthy nervous system be with you.